This is the first of two videos on the urinary system, and we're going to start out by taking a look at the gross anatomy of the kidneys. The kidneys are retroperitoneal. They're in the superior lumbar region, located between vertebrae T12 and L5. The right kidney is crowded by the liver, so that's going to rest a little bit lower than the left kidney will. The adrenal or suprarenal gland sits on top of each kidney. The kidney has a convex lateral surface and a concave medial surface with a renal hilum that leads to the internal space, which is called the renal sinus. We'll take a look at that in more detail in just a minute. At the hilum, you have the entrance of the ureters, the renal blood vessels, the lymphatics, and the nerves. And I'm sorry, that would be the exit of the ureters, and entrance and exit of the blood vessels, lymphatics, and nerves. This is the position of the kidneys against the posterior body wall. You can see again that they're covered slightly by the 12th rib. There are three layers of supportive tissue around the kidney: the renal fascia, which is the anchoring layer of dense fibrous connective tissue; the perirenal fat capsule, which basically means beside the kidneys. This is a fatty cushion for the kidneys. The kidneys don't have the protection of the fluid in the peritoneum that the abdominal organs do, so a little bit of extra fat is necessary here. And the fibrous capsule, which is a transparent capsule that prevents spread of infection from locations in the abdominal cavity to the kidney. This is the position of the kidneys against the posterior body wall. This is a transverse section across the body, and we were actually looking up at the kidneys. So that muscular layer that you see is actually the diaphragm. So that's the diaphragm right here and here. And what we're going to do is take a look at the three layers that actually surround the kidney. So here's the anterior renal fascia, the posterior renal fascia, the perirenal fat capsule, and the fibrous capsule. So we're going to talk about a homeostatic imbalance based on the location of the kidneys and impact. So the upper parts of both kidneys get protected by the thoracic rib cage. We've seen that in a prior slide. And also the perirenal fat provides cushioning. However, the lower parts of the kidneys are susceptible to blunt trauma, especially the right kidney, because as you'll recall, it kind of gets crowded out by the liver. And examples of blunt trauma would be falls, motor vehicle accidents, or contact sports injuries. The renal artery is really vulnerable to injury from any kind of rapid deceleration, and this can be, again, during car crashes. This can lead to lacerations in which the renal artery is torn, or a thrombosis, a blood clot. And so this is something that can also happen in contact sports injuries. And what's the sign that this has happened? It's when you find hematuria, which is blood in the urine. This is an important sign of this kind of trauma. And it's something that I heard discussed as a freshman football player about one of the varsity players at one point. So surgical treatment may be required, not always, but sometimes, depending on how severe the injury to the renal artery is. The internal kidney has three distinct regions: the renal cortex, which is the most superficial region, and it appears somewhat granular under a microscope; the renal medulla, which is deep to the cortex. This is composed of medullary or renal pyramids. The broad portion of the pyramid faces the cortex, and the papilla, which is the tip of the pyramid, points internally towards the renal pelvis. The renal pyramids are separated by renal columns, and these are inward extensions of the cortex, of the cortical tissue. There are lobes in the kidney which consist of the medullary pyramid and its surrounding cortical tissue, and there are about 8 lobes per kidney. The third section layer is the renal pelvis. This is a funnel-shaped tube that is continuous with the ureter. It's composed of minor calyces, which are areas that collect urine draining from pyramidal papillae, and major calyces, which collect urine from the minor calyces. So we'll see this in more detail in just a minute. The major calyces will empty the urine into the renal pelvis, and urine flow will proceed from the renal pyramids to the minor calyx, to the major calyx, to the renal pelvis, to the ureters. So here's the internal anatomy of the kidney, and we're going to go from the outside in. So first you're going to see the fibrous capsule. Inside the fibrous capsule, you'll see the layer, the outermost layer, which is the renal cortex. Deep to the renal cortex, you'll see the renal medulla, and the cortex will actually extend down into the medulla. These are these sections of the cortex called the renal columns. The renal columns surround the renal pyramids in the renal medulla. And these are the papillas of the pyramids, so the sort of pointed parts at the base of the pyramids that lead into the minor calyx, or calyces is plural. The minor calyces will themselves lead into major calyces, so what I want you to notice here is that the major calyx has got more than one minor calyx draining into it. The major calyces will drain into the renal pelvis, and then the renal pelvis, as you can see to the right, to its right, drains into the ureters. So in this slide we're going to talk about kidney infections. So pyelitis is an infection of the renal pelvis and the calyces, while pyelonephritis is an infection or an inflammation of the entire kidney. Infections in women are usually caused by fecal bacteria entering the urinary tract, and severe cases can cause swelling of the kidney and abscess formation, and in these cases pus may actually fill the renal pelvis. If this is left untreated, then you can get pretty severe kidney damage. Normally this is successfully treated with antibiotics. So in the prior slide I mentioned where some of these kidney infections might come from, they usually work their way up the ureters to the kidneys. And so the ureters enter the kidneys at the hilum, so at this point it makes some sense to talk about the blood and nerve supply, which also enters and exits at the hilum of the kidney. So kidneys are responsible for cleansing the blood and adjusting its composition, and it means that they've got a very rich blood supply. Renal arteries deliver about 1/4 of the cardiac output to the kidneys each minute. So the arterial flow goes from renal arteries to segmental to interlobar to arcuate to cortical radiate, which are also called interlobular. So we'll see a diagram where we see each of these. Venous flow is going to be cortical radiate to arcuate to interlobar to renal veins. So one of the nice things about memorizing the blood supply of the kidneys is that the arteries and the veins parallel very well. So once you learn the path in, you've pretty much learned the pathway out. So there are no segmental veins. Again, so we'll see this in a future diagram. And the nerve supply is via sympathetic fibers from the renal plexus. And again, this is actually going to enter and exit through the hilum of the kidney. So we're going to take a look at the blood supply now, and let's orient ourselves. You can see the ureter and the renal pelvis. You can also see the two main layers of the kidney, the renal medulla and the renal cortex. So entering the kidney, blood will enter at the renal artery. From there, it will branch into segmental arteries. Those segmental arteries will branch into interlobar arteries, which usually go through the columns, the renal columns that we've already talked about. Now, once these interlobar arteries reach basically the border between the cortex and the medulla, they're going to kind of turn and go into an arc around the pyramids. And so these are called arcuate arteries after their shape. The arcuate arteries will send branches off that are called cortical radiate arteries. These are also called interlobular, but to be honest, I prefer cortical radiate because they're located in the cortex and they radiate out. So cortical radiate artery is a name that makes a whole lot of sense to me. So from the cortical radiate, they'll go through the nephron. We'll discuss that blood pathway in the next video. So from the cortical radiate artery, you'll go into the nephrons, and then you'll start your journey back through the cortical radiate veins. Those cortical radiate veins will merge into arcuate veins. Arcuate veins will merge into interlobar veins. And as I mentioned before, there are no segmental veins, so the arcuate veins will themselves drain into the renal vein, which exits the kidney. Okay, so in this slide, we're going to talk about the path of blood flow through the renal blood vessels. And what's different about this from the prior slide is that we're also going to discuss microscopic blood vessels that are associated with the units of filtration in the kidneys, which are called the nephrons. And a little later on in this slide, we're going to see diagrams of the nephrons themselves, and so this should be a little bit more clear when you see those. So the renal artery is a branch off of the abdominal aorta. So the abdominal aorta gives rise to the renal artery. The renal arteries will give rise to the segmental arteries, which themselves give rise to the interlobar arteries. The interlobar arteries give rise to the arcuate arteries, which, as we saw in the previous slide, are right at the border of the cortex and medulla of the kidney. So these arcuate arteries will give rise to cortical radiate arteries, and at this point, the blood vessels are going to be small enough to where we're talking about blood vessels that are associated with the nephron. So each of these blood vessels that are within the dotted lines here are nephron-associated blood vessels. So the vessel that brings blood into the nephron is the afferent arteriole. The afferent arteriole will give rise to the glomerulus, which is a specialized ball of capillaries. The glomerular capillaries will themselves merge back into the efferent arteriole. So this is a rare case in which you see an arteriole give rise to capillaries, which give rise to another arteriole. And what that should tell you is that there's no gas exchange occurring in this capillary bed. The efferent arterioles will then branch into the peritubular capillaries, or if you're talking about a juxtamedullary nephron, the vasa recta. And again, we will see pictures of this in just a little bit. So in this case, you can see from the purple color here, there's going to be some gas exchange going on in these peritubular capillaries. So the peritubular capillaries will then combine back into the cortical radiate veins, which combine back into the arcuate vein. And at this point, I want to make this clear that the peritubular capillaries can or the vasa recta can, the vasa recta can go back into the cortical radiate vein or into the arcuate vein. So the arcuate veins will drain into the interlobar vein, which drain into the renal vein, which will drain into the inferior vena cava. Before going any further, let's take a look at the nephron itself. So the nephron is the functional unit of the kidney. There are several million in each kidney. And the nephron is actually responsible for filtration of blood plasma into the filtrate that you find in the kidney. It's also responsible for secretion into the urine. It's responsible for reabsorbing substances from the urine. So tubular filtration, secretion, and reabsorption, these are all processes that happen in the nephron. And so most nephrons, about 70% of nephrons, are actually entirely contained in the renal cortex. About 30% of nephrons, called juxtamedullary nephrons, are actually send loops down into the medulla. So let's take a look at one of these juxtamedullary nephrons and examine the different parts of it. So first you have the glomerular capsule, which surrounds the glomerulus. So the glomerulus is a specialized ball of capillaries, which we'll get into in a lot more detail in just a minute here. So blood flows through the glomerulus. You get leakage of blood plasma and contents of blood plasma out into the glomerular capsule, and this filtrate that leaks out of the glomerulus will enter the tubular system of the kidney. So the tubular system of the nephron actually begins with the proximal convoluted tubule. So the proximal convoluted tubule gives rise to the descending limb of the nephron loop. Now, for ease of seeing the different parts of the nephron here, the nephron loop is turned in a way that it normally doesn't turn. So the nephron loop goes down and continues to the left on this diagram. In a juxtamedullary nephron, the loop would actually turn back to the right, and the last portion of the loop, you'll go up the ascending limb of the nephron loop, but following that you've got the distal convoluted tubule, which will actually be right back next to the glomerulus, and I've got a better diagram of this a little bit later on that shows this. So this is the nephron. In the glomeruli of the kidneys, the volume of fluid that is filtered is larger than in most capillaries of the body, and there are a number of reasons for this. First of all, There's a larger surface area for filtration than what you're going to see in most capillary beds. And so with more surface area for fluid to leave, more fluid will leave. The filtration membrane itself is thin and porous, and we'll go into the details of the filtration membrane in a later slide, but it is thin and it is more porous than it is in most capillary beds. The glomerular capillary blood pressure is higher than the pressure in other capillary beds. And the main reason for this is because the efferent arteriole, which you see here, the diameter of it, if you look up and to the left, it's smaller in diameter than the afferent arteriole here. And so basically having that smaller diameter of the efferent arteriole, it's kind of like putting a crimp in a hose. Upstream of the crimp, you're going to have higher pressure. And so upstream of this sort of crimp in the efferent arteriole, you have higher pressure because of the difference in the diameters between these two arterioles. Also, you have mesangial cells indicated by the arrows here. Now, there are going to be some that are located in the glomerulus itself, but they're also located, as shown by these little green arrows, around the afferent arteriole. So if these cells are relaxed, they result in a higher filtration rate because when they're relaxed, they're not clamping down on the afferent arteriole and the flow in is relatively high. So when these mesangial cells contract, what they're going to do is they're going to narrow the afferent arteriole. And this will result in a lower filtration rate because if you narrow the lumen of the afferent arteriole, you're going to decrease the amount of blood flow coming in, and therefore your filtration rate will drop. So let's take a look at the filtration pressure itself. So there are a number of forces that determine exactly how much fluid gets filtered out of the glomerulus. So the first is glomerular blood hydrostatic pressure. So this is hydrostatic pressure that begins with the left ventricle of the heart. This is the basically your arterial blood pressure. So this is going to be a force that pushes blood plasma out of the capillaries. This promotes filtration, which is, and again, I want to make it clear that filtration is the process of fluid going through the membranes of the glomerulus out into the Bowman's capsule. So this hydrostatic pressure promotes filtration, and it's a force of about 55 millibars. Capsular hydrostatic pressure is present in this case, and so because the glomerular capsule is as small as it is, it's going to resist this hydrostatic pressure of the glomerulus with its own hydrostatic pressure that kind of pushes back. And so it'll actually push back at a rate of about 15 millibars. So this is something that opposes filtration. It doesn't encourage filtration, it opposes it. The third force here is blood colloid osmotic pressure. And this is where some of the proteins of the bloodstream will exert osmotic pressure. And what this does is it pulls fluid back into the glomerulus. And this also then opposes filtration. And it happens at about 30 millibars. And so what is your net filtration pressure? Your net filtration pressure is going to be the glomerular pressure pushing out. It's that pressure pushing out will be taken away from by the osmotic pressure and by the capsular hydrostatic pressure. So how do you calculate the net filtration pressure here? This is how net filtration pressure gets calculated. The net filtration pressure is going to be the glomerular hydrostatic pressure minus the capsular hydrostatic pressure and the blood osmotic pressure. So basically here you've got the one force that pushes out, you've got the other forces which are trying to push fluid back into the capillaries. So your net filtration pressure in this case is going to be the glomerular hydrostatic pressure, which you can see highlighted as 55 millibars. So this is the pressure that's pushing out. The pressure that's trying to bring fluid back into the glomerular capillaries is the blood colloid osmotic pressure. The capsular hydrostatic pressure, which is at 15 millibars, and the blood colloid osmotic pressure, which is 30 millibars. And so if you take 15 plus 30, which is 45, if you take that away from 55, you're going to have a net filtration pressure of 10 millibars, which you can see right here in the red circle. So the glomerular filtration rate, or GFR, is the amount of filtrate formed in all of the nephrons of both kidneys each minute. And as you might imagine, the glomerular filtration rate is very tightly regulated. The glomerular filtration rate is directly related to the net filtration pressure. So basically, if you have something that alters one of these forces, perhaps alters the glomerular hydrostatic pressure, then you're going to have an alteration in net filtration pressure, which will then alter the glomerular filtration rate. So if your body's blood pressure drops, then the glomerular hydrostatic pressure drops. This will result in a drop of the net filtration pressure. So let's just imagine for a minute here that the glomerular hydrostatic pressure drops from 55 to 50. The blood colloid osmotic pressure is not going to drop, and neither will the capsular hydrostatic pressure. So let's say you drop your glomerular hydrostatic pressure from 55 to 50, then in this case you would have 50 minus 45, which would give you a net filtration pressure of 5 millibars. And with a lower net filtration pressure, your glomerular filtration rate will also drop. So filtration will actually halt if the blood hydrostatic pressure drops below 45 millibars. And the reason why is because your blood colloid osmotic pressure is going to remain constant. It's always going to be 30 unless you're under some sort of radically advanced starvation or some other situation where your blood proteins are really reduced. Okay, so that blood colloid osmotic pressure is going to remain constant. The capsular hydrostatic pressure, the capsule isn't going to change shape or size, so its pushback pressure, that hydrostatic pressure, is always going to remain at 15. So no matter what your glomerular hydrostatic pressure is, you're always going to have an opposing force of 45 millibars. So that means that if your glomerular hydrostatic pressure drops, the purple arrows again, drops below 45, the opposing forces are too much for any fluid to leave the capillaries, and it just continues on through the circulatory system. So filtration will halt if the blood hydrostatic pressure drops below 45. Now, GFR is constant at mean arterial pressures of between 80 to 180 millibars. So what that means is that it takes an unusually high or an unusually low blood pressure to alter your glomerular hydrostatic pressure. There are regulatory mechanisms for glomerular filtration. So basically, what you want to try to do is you want to try to keep your filtration rate from being too high or too low. So you can regulate your glomerular filtration rate by adjusting blood flow into and out of the glomerulus. And so an increased blood flow will result in increased filtration, which results in an increased filtration rate. If you vasoconstrict the afferent arteriole, this decreases blood flow and decreases filtration and decreases filtration rate. So if you'll recall, the efferent arteriole is smaller in diameter than the afferent arteriole. If you vasoconstrict the afferent arteriole, there is not as big of a difference between the two diameters, and therefore it's like easing up on a crimp in a garden hose, so the pressure isn't going to be quite as high. So if you vasoconstrict the afferent arteriole, you decrease blood flow, you decrease glomerular filtration rate. Vasodilation, so if you open up that afferent arteriole bigger, then you will increase blood flow. So this would be like turning, basically, in our garden hose model, somehow making the hose a little bit bigger. So basically, if you increase blood flow into the glomerulus, you're still going to have that crimped down efferent arteriole, and increase in blood flow will increase the glomerular hydrostatic pressure. It'll increase the filtration above 10 millibars. So look at your diagram. Let's say that you increase your glomerular hydrostatic pressure to 60. that would increase your net filtration pressure to 15 millibars and thereby increase the rate at which filtration occurs throughout the entire kidney. So you can accomplish this by vasodilating the afferent arteriole. If you alter the glomerular capillary surface that's available for filtration, this can also help regulate glomerular filtration. So there are three mechanisms that are going to control GFR. The first is called renal autoregulation, the second is neural regulation, and the third is hormonal regulation, and what we're going to do is to examine those one at a time. So there are two components to the renal autoregulation. First of all, there's something called the myogenic mechanism, and secondly, there's something called tubulo-glomerular feedback. Now, along with hormonal controls, these maintain adequate filtration over a wide range of systemic blood pressures. So you'll recall that I mentioned just a little bit ago that the net filtration rate remains constant between 80 and 180 with a mean arterial pressure. And this is a really wide range of systemic blood pressures. So basically, if blood pressure goes up or down, then these mechanisms can respond to changes in blood pressure and keep your glomerular filtration rate relatively constant. So let's take a look at the myogenic mechanism first. And before we take a closer look at the glomerulus itself, I wanted to show you the angle that we're looking at with the glomerulus. So this is a picture of a nephron, and what you can see here is this one is diagrammed a little bit more correctly with the distal convoluted tubule coming back up right beside the glomerulus. And so basically if we cut through both the distal convoluted tubule and the glomerular capsule right at this angle, we're going to see the glomerulus and we're going to see the distal convoluted tubule. So let's fade that out and take a look at this. Okay, so if the walls of the afferent arteriole stretch, this is going to cause the smooth muscles that are in the afferent arteriole to contract. So basically, like any other arteriole, the afferent arteriole has smooth muscle in the vessel walls. And so if the walls get stretched out, then you're going to see these smooth muscles contracting as a response. So right here, this is called the myogenic mechanism, basically meaning that this mechanism is carried out by muscle or myo. Now the contraction of these smooth muscles will decrease the diameter of the afferent arteriole. So that means if your systemic blood pressure rises and increases the blood flow in, you're going to clamp down and decrease the diameter of the afferent arteriole and reduce the amount of blood flow that would have otherwise been coming in. So closing down the diameter of this arteriole is basically going to offset an increase in blood pressure. Now, it's vice versa when the systemic blood pressure drops. So if the systemic blood pressure drops, if you go into lower blood pressure, then the afferent arteriole will actually expand. And this happens because the smooth muscles in the afferent arteriole will actually relax under less pressure. So this myogenic mechanism can help compensate for changes in systemic blood pressure. This is something that works within seconds, so this is a very quick response. So this is a sort of a zoom in on the renal corpuscle diagram that we saw earlier. And let's take a little bit of a closer look at this diagram and see exactly what we're going to be looking at. So this is the afferent arteriole. These are the juxtaglomerular cells. This is the endothelium of the glomerulus. These are the mesangial cells. These are podocytes, which is a different type of cell that we haven't talked about before. These are cells that overlie the capillaries of the glomerulus. These podocytes have cytoplasmic extensions called pedocells. This is the capsular space between the glomerulus and the borders of the Bowman's capsule. And this is the parietal layer of the Bowman's capsule. This is the proximal convoluted tubule. And finally, this is the efferent arteriole. This is the ascending limb of the loop of Henle. And this is the macula densa. This is also a set of mesangial cells, and it's the contraction of these mesangial cells which can contract the afferent arteriole. Next, we're going to talk about tubuloglomerular feedback. And basically, this is the idea that what's happening in the tubules themselves can actually feed back to the glomerulus. This is the reason why this diagram shows the distal convoluted tubule right next to the glomerulus. So if you see this portion right here, this is actually the distal convoluted tubule here and here, all the way around here. And so you can see that it borders the glomerulus, and what happens is that this sort of juxtaposition allows the tubule to give feedback to the glomerulus. So the macula densa that's in the last portion of the loop of Henle, or the beginning of the distal convoluted tubule, will provide feedback to the glomerulus. And this is the macula densa. It's that group of cells indicated by the green arrows. So if systemic blood pressure gets elevated and it manages to overcome, let's say, the myogenic mechanism, then with a greater amount of filtration, it's going to speed up the flow of fluid in the tubular system. Now, this increase in flow of the tubular system means that you don't have time to reabsorb what you need to reabsorb from the filtrate. So there's less time for proper reabsorption of sodium chloride and water. So this would be a good time to say that when you see filtration of materials out of the glomerular capillaries, you don't have just wastes leaving. You also have sodium, you have chloride, you have water, you have glucose. You have a lot of things that the body would like to reclaim. And so part of what happens in the tubules, the distal convoluted tubule, the proximal convoluted tubule, the loop of Henle, part of what happens is reclamation of things that you need to bring back into the bloodstream. Now, if there's too high a rate of flow through those tubules, then you've got less time for proper reabsorption of sodium chloride and water, and that means that more of it is going to exit the body during urination. And so basically you don't want fluid flow through these tubules to be too high so that you can get proper reabsorption of what you need to reabsorb. So basically the macula densa will detect this increased fluid flow and inhibit nitric oxide release from juxtaglomerular cells, which are located here. So without this vasodilator, without the nitric oxide, vasoconstriction will occur, and this decreases blood flow into the glomerulus. So basically the mechanism works like this. So if you have nitric oxide release from juxtaglomerular cells, then you're going to see dilation of that afferent arteriole, increased blood flow into the glomerulus, increased filtration rate. Now, let's say again that fluid flow through the tubules is too fast, too much filtration is going on. What the macula densa will do is to cut down on the vasodilator. It means that the afferent arteriole doesn't get vasodilated, and this will decrease the blood flow into the glomerulus, drop your filtration rate. So this is something that's going to work slower than the myogenic mechanism. The third thing is going to be sympathetic nervous input to the blood vessels of the kidney. So this sympathetic input is basically norepinephrine. At rest, there's only a small input of norepinephrine and renal autoregulation. So basically the local mechanisms, meaning the myogenic mechanism and the tubuloglomerular feedback, which we've just looked at, the renal autoregulation under normal circumstances predominates and is responsible for regulating glomerular filtration rate. However, when the sympathetic nervous system begins to put input into here, both the afferent and the efferent arterioles will constrict. So, with moderate input by the sympathetic nervous system, glomerular filtration rate only decreases slightly. However, if you have strong input for the sympathetic nerves, then the afferent arteriole will constrict more than the efferent. And again, with decreased blood flow into the glomerulus, this is going to drop your filtration rate. So your glomerular filtration rate can get greatly decreased with a constriction of afferent arterioles. Now you're going to see this when blood is needed in other areas of the body. So basically, if you've got times of shock due to hemorrhage, dehydration, stress, or if you're in a fight or flight situation, then you're going to need blood flow to areas other than the kidneys, because at that point, urination may not be the most important thing that the body can do. You may need blood being rerouted to skeletal muscle. So you're going to see this neural regulation of GFR under abnormal circumstances. Okay, so the last thing that we're going to talk about in terms of regulation is hormonal regulation of tubular reabsorption and secretion. There are a number of different mechanisms that work to accomplish this. The renin-angiotensinogen-angiotensin mechanism, aldosterone, antidiuretic hormone, and atrial natriuretic peptide. Now, some of these we have made reference to in previous chapters, basically the endocrine system, but we'll talk about these again in this video. So basically, with respect to the different mechanisms that we've seen the myogenic mechanism of autoregulation, which basically happens when, if your systemic blood pressure drops, you're going to have decreased stretch of the smooth muscle in the walls of the afferent arterioles, and they'll vasodilate and increase your GFR. So if you have a decreased GFR, you have a decreased filtrate flow and decreased sodium chloride in the loop of Henle. And basically what's going to happen is the macula densa cells, again, are going to inhibit vasoconstriction. This is going to cause vasodilation of afferent arterioles, and this is going to result in a higher GFR. Our neural controls we've already seen. And again, baroreceptors in blood vessels of systemic circulation will send signals back to the sympathetic nervous system, and this is going to actually play into what happens in the hormonal mechanism, which we're going to take a look at right now. So in the hormonal mechanism, granular cells in the juxtaglomerular apparatus of the kidney, which we've seen in that prior video or prior slide, they're going to release renin. And what renin will do is to basically start a cascade that results in the conversion of angiotensinogen on down the line to angiotensin II. Now what angiotensin II will do is to cause systemic, not the afferent arterioles, but systemic arterioles to vasoconstrict. This will increase peripheral resistance, which will increase systemic blood pressure. That will lead to a higher glomerular filtration rate. In addition, what angiotensin II will do is stimulate the adrenal cortex to release aldosterone. This targets kidney tubules and has them reabsorb more sodium. Now, when they reabsorb more sodium, water will follow, and as a result, the volume of fluid in the bloodstream will actually rise, and this will cause systemic blood pressure to rise. So all of these are responses to systemic blood pressure being lower, which is a condition that can be fatal fairly quickly. Next, we're going to talk about filtration, tubular secretion, and reabsorption in the nephrons. Here's a more detailed look at the filtration membrane itself. So we're going to go from the inside out basically with this. So the first part of the filtration membrane is fenestration or pores of the glomerular endothelial cells. So when we talked about the circulatory system earlier this semester, we talked about three types of capillaries. Continuous, fenestrated, and sinusoid. So these are going to be fenestrated capillaries which have pores in the cells. So this is going to prevent filtration of blood cells. In other words, blood cells won't be able to get out, but it will allow all other components of the blood plasma to pass through. The next layer in gray here is the basal lamina of the glomerulus. And so this is a protein layer that surrounds the glomerular capillaries, and this is going to prevent filtration of larger proteins. Finally, we have the slit membrane between the pedicels, and this is going to prevent filtration of medium-sized proteins. So again, none of the formed elements of the bloodstream, any types of blood cells or platelets, should be present in the urine because of the first part of the filter, and there shouldn't be any proteins present in the urine because of the second and the third portion of this filtration membrane. Here's a diagram which is going to show us two of the three components. That's why I didn't start out with this one. But this diagram shows us two of the three components of the filtration membrane. First of all, you see the glomerular capillary endothelium, and in this case, the basement membrane is removed and the podocyte covering is removed. And what you basically get to see here is that there are fenestrations or pores in the endothelium itself, and this is going to allow these capillaries to be more porous than most capillary beds. So let's talk about the podocytes themselves in a little bit more detail. This is the reason why I basically wanted to include this slide. So here's the cell body of the podocyte, and basically what it's going to do is it's going to send cytoplasmic extensions out, and these cytoplasmic extensions are then going to divide into what are called the foot processes of the podocyte. So podocyte itself literally means foot cell, and I guess somebody thought that these look like feet, but these are the processes of the podocyte. Now you'll notice that these don't completely cover the glomerular capillary endothelium. So they don't cover it. Instead, what they have is filtration slits that are in between these foot processes. So if the podocyte changes the shape of its foot processes and reduces the space of the filtration slits, you're going to reduce the surface area that's available for filtration because they would be covering up um, the other filters. So if these foot processes expand, then the filtration slits narrow and there's less filtration. What all of this leads into is the three main processes that occur in the nephron. Glomerular filtration, in which water and solutes leave blood plasma and move into the tubular system. Tubular reabsorption, in which 99% of this filtered water and solutes get reabsorbed back into the blood plasma of peritubular capillaries and the vasa recta. And tubular secretion, in which secretion of wastes, drugs, excess ions into the tubular system. These get secreted into the tubular system independently of hydrostatic pressure. And so these substances will definitely be excreted into the urine. The major site of reabsorption and secretion is the proximal convoluted tubule. The solutes that get reabsorbed here are sodium, potassium, calcium, chloride, bicarbonate, and phosphate. Substances that actually get secreted here can include hydrogen ions, potassium, ammonium, creatinine, and some drugs, including most notably penicillin. So these are the substances that are filtered, reabsorbed, and excreted in the urine. And as you can see, most of these are actually handled by the proximal convoluted tubule. So how does reabsorption then occur? So reabsorption occurs via two routes. First of all, paracellular reabsorption, which occurs between adjacent tubule cells. So basically what this means is that even though these tubule cells are joined by tight junctions, some fluid can still leak between the cells. The other is transcellular reabsorption, which means that materials actually pass through tubular cells on their way back to the capillaries. So basically substance will pass from the lumen of the tube through the apical surface of the epithelial cell. So this is the surface that's going to face the lumen. So basically, substances will go through this apical surface, then they'll go through the cytosol of the tubular epithelial cell. They'll go out of the basolateral membrane into interstitial space and back into the bloodstream via the peritubular capillaries. And we'll have a diagram in just a minute that should make that make a little bit more sense. Let's take a little bit closer look at these routes for reabsorption. And so the transcellular route is going to involve a number of different steps here. Before we get started with that, I want you to notice that there are two different colored arrows here. One is going to be passive transport. So water will be able to passively transport across the luminal membrane. The other is active transport. So most of the solutes that actually are pushed out during the filtration process cannot diffuse back across the membrane on their own, so they have to be assisted with active transport. So the first part of movement via the transcellular route is going to involve transport across the luminal membrane. So right here, so you can see that both solutes and water have to pass across the luminal membrane. The next thing that happens is diffusion through the cytosol. So you'll notice that one of those green arrows remains, and what I wanted to show you with that arrow is that sometimes the solutes will actually join into the paracellular route, which is just beneath it. So the third thing that happens is transport across the basolateral membrane, which you'll see here. And then the fourth thing that happens is movement through the interstitial fluid and into the capillaries. So basically this is how H2O and how solutes are going to use the transcellular route. The next thing that we'll look at is the paracellular route, and so we've got H2O just moving between the cells, in between the cells, and eventually into the peritubular capillary. And the same thing can happen for solutes. They can move between cells and thus into the peritubular capillary. So when renal cells transport solutes in or out of tubular fluid, they usually move specific substances in one direction only. Now you're going to find different types of transport proteins in the apical membrane, which again faces the lumen, versus the basolateral membrane, which faces the peritubular capillaries. And sodium would be the best example of this. Sodium is going to be primarily found in the extracellular fluid space. There's actually a low concentration in the tubular cells themselves. And so what this means is that there's going to be a higher concentration of sodium in the tubular fluid, which you see at the top of this diagram in yellow, and in the interstitial fluid that surrounds these cells, which is in sort of that lighter pink, so the peritubular fluid. There's a higher sodium concentration in these fluid spaces than there is in the actual cells, which are diagrammed here in purple. And so the reason why this happens, why there's a low concentration of sodium in the cell itself, is because there's sodium-potassium ATPase pumps in the basolateral membrane here that are going to move sodium from the cell downward out towards that peritubular fluid. There are no sodium-potassium ATPase pumps in the apical membrane here. So basically what happens is your flow of sodium is unidirectional because the pumping of the sodium in this direction at the basolateral membrane means that there's going to be a difference in concentration between the cells and the tubular fluid. And so basically, sodium ions will come through carrier proteins down their concentration gradient across the apical membrane in this direction. There are other substances that use a similar mechanism, and so basically glucose will pass through the membrane via carrier proteins. And sometimes when these sodium ions come through, they can use, other substances can use an antiport or countertransport pump to move substances into the tubular fluid, and that's how secretion can occur. This is a different look at tubular reabsorption. It's got a little bit more detail on this slide. So the first thing let's do is get oriented. So in this case, the apical surface of the cell is on the left, and the basolateral surface is on the right. So that means that this yellow fluid that you see on the left is going to be your tubular fluid, and the peritubular capillary is going to be on the right. So now that we're oriented, let's kind of get started with this. So at the basolateral membrane, and look to the right of the cell, sodium gets pumped into the interstitial space by the sodium-potassium ATPase pump. And so that's right here. And so basically what it's going to do is it's going to pump out 3 sodiums out into the interstitial fluid, 2 potassiums in. So what is it that actually happens to the potassium and the sodium? Well, the potassium will actually leave the cell again almost immediately through carrier proteins, and it will be available to be brought back into the cell as part of the action of this pump. The sodium that's brought out into the interstitial fluid in the meantime is actually going to go into the peritubular capillaries, and it's not going to be available anymore And so what that's going to do is that's going to cause a concentration gradient where you have a lower concentration of sodium within this tubule cell than you have in the tubule lumen. And what's going to happen then is that sodium will come from the tubular fluid into the tubule cell here. And so you'll notice that this is a dashed line at the bottom that says secondary active transport. So basically your active transport with the sodium potassium pump has enabled a concentration gradient, and so sodium just comes right through this channel on the apical membrane. So when the sodium comes in, then sometimes you can get reabsorption of some organic nutrients and ions by co-transport here. So at the same time that the sodium comes back in due to this concentration gradient, then you'll have glucose, amino acids, some ions, and some vitamins coming in right along with the sodium. So it's this sodium-potassium pump at the basolateral membrane which sets up the co-transport of glucose, amino acids, some ions, vitamins, and this is how their reabsorption will actually happen. How does H2O get reabsorbed? It can get reabsorbed via either osmosis, or it can be reabsorbed through its own channels, as you can see here. Okay, now water reabsorption does increase the concentration of the solutes that are left in the tubule filtrate. So then these solutes can be reabsorbed as they move down their concentration gradients. And so if substances are lipid soluble, then they're going to be able to cross cell membranes without any assistance, and so you'll see these lipid soluble substances moving in. You will see some ions and urea moving by the paracellular route, so in between cells right here. So this is how a lot of the reabsorption occurs. So the main driver here for things like glucose and amino acids and some ions is this sodium potassium pump setting up a concentration gradient that sodium will then come through at the apical membrane and bring these other substances right along with it. Okay, so the primary active transport is the sodium potassium ATPase pump, and we've talked about that a great deal in AMP1, so you basically know what's going on there. But the transport mechanism that the ATPase pump sets up is the secondary active transport. And so again, the secondary active transport results because you've set up a lower concentration of sodium in that tubular cell. Okay, so in the proximal convoluted tubule, which is what we've been talking about, the largest amount of solute and water reabsorption occurs in the proximal convoluted tubule. And as I indicated in the earlier slide, most of these absorptive processes actually involve sodium. There are two different types of sodium transporters. So again, this reabsorption process occurs a lot with symport with sodium. And what are the things that usually get symported back in with sodium? And again, remember that you've got these substances coming in along with sodium because of that lower concentration of sodium in the tubule cells. So you're going to see glucose, phosphate, sulfate, amino acids, lactic acid, some vitamins. These things will be symported in with sodium. And going back to our prior slide, let me just remind you that this symport happens along with sodium. So you're going to have antiporters as well. And with these antiporters, you're going to see sodium and hydrogen antiporters. And so as sodium comes into the cell, which we've seen in a couple of different slides, hydrogen won't be brought along with it. Hydrogen will actually go in the opposite direction. Hydrogen gets secreted. And what's the effect of this hydrogen secretion? Well, there's a conversion that is actually happening in the tubular fluid at this point. You have CO2 and H2O being combined to carbonic acid. And this is going to result in bicarbonate, which is HCO3. So as this is happening out in the tubular fluid, what this means is that when you have a hydrogen ion secreted into the tubular fluid, this is going to cause reabsorption of a filtered bicarbonate ion. So what substances get reabsorbed? Sodium, cations, chlorine, bicarbonate, water, lipid-soluble solutes, urea, and small proteins. So this is kind of our summary of what happens in the proximal convoluted tubule. So some things that I want you to notice here is that the glucose and the amino acid reabsorption is incredibly efficient. You're going to reabsorb 100% of your glucose. You're going to reabsorb 100% of your amino acids. And so the only instance under which you're going to see glucose or amino acids in the urine is if there is an incredibly high amount of either glucose or the amino acids in the bloodstream. And this is something that is probably happening with something else is going on here. It could be either an unusually large ingestion of glucose or amino acids, or it could be that somebody's diabetic in the case of glucose. So you should never see glucose in the urine. Bicarbonate, the reabsorption is almost as efficient. With water, sodium, potassium, and chloride, not quite as efficient, and you're going to see a little bit of reabsorption in later portions of the nephron tubules. The amounts of calcium, magnesium, and phosphate, this kind of varies basically based on the body's needs. Now, when you enter the loop of Henle, your filtrate is quite different than the filtrate at the beginning of the proximal convoluted tubule. But because you've absorbed approximately the same percentage of water and the overall percentage of solutes, the osmolarity of the fluid is essentially the same. Because the proximal convoluted tubule reabsorbed water and solutes in equal proportions. So in terms of the loop of Henle, the substance that gets reabsorbed is going to be water in the descending limb and ions in the ascending limb. And so the descending limb is going to be right here. And so reabsorption of water means that water is going to be leaving the ascending limb here. Now it's leaving by osmosis, which means the further, the deeper and deeper that you get into here, you're still going to see some water leaving. And so this portion is impermeable to the solutes. And so that means that osmosis is going to continue to drive water out of the descending loop of Henle as you move down it. Now, when you get back up into the ascending limb, then you're going to see the reabsorption of ions out here at the ascending limb. And the reason why you're going to see reabsorption of ions in this ascending limb is because it is actually impermeable to water. And we're going to examine that in more detail in our next video. Okay, so in the loop of Henle, again, what do you get reabsorbed? 20 to 30% of sodium, potassium, and calcium. The remaining bicarbonate, 35% of chloride, and 15% of water. And so basically what I wanted to show you is that in the ascending limb here, so if you look at this, what it says right here, it says that this plasma membrane is impermeable to water. And what you're going to see happening here is the same basic kind of mechanism that you saw in the proximal convoluted tubule, where you have chlorine and sodium moved out into the peritubular fluid across the basolateral membrane. And this is going to set up a concentration gradient which will bring chlorine and sodium in through the apical membrane here. So basically, again, your basolateral membrane is kind of going to be driving what happens at the apical membrane. Now, water reabsorption is not linked with the absorption of solutes here because, again, this plasma membrane is impermeable to water. So in most cases, when you see solutes move, you're going to see water following by osmosis. But because this particular portion of the loop of Henle is impermeable to water, then you're not going to see water moving along with it. Next, we come to the distal convoluted tubule, and we're going to see continued reabsorption of sodium and chloride here. You're also going to see about 10 to 15% of water reabsorption, but what may be most important is that this is the site where parathyroid hormone stimulates reabsorption of calcium. So how does a lot of this kind of thing happen? Well, principal cells will reabsorb sodium and secrete potassium. So basically, sodium will enter the apical portion of tubular cells by leakage channels, but the most important thing here is that they are actively transported out of the cell by the basolateral membrane at the sodium-potassium ATPase pump. So what you see happening here, see this sodium-potassium pump driving down the sodium concentration. And so you're going to see transport proteins at the apical membrane bringing sodium in and secreting potassium. Same basic thing is going to happen with hydrogen. So as you have a sodium hydrogen ATPase pump, and so that's going to drive sodium out. It's also going to bring hydrogen in. And so again, you're going to see at the apical membrane antiport of sodium and hydrogen. Now, although the arrow doesn't show the sodium coming in on this next thing, this is also where you find carrier proteins that are going to secrete toxins and drugs, and they're also going to be antiported out in opposition to sodium. So basically, sodium and chloride will get reabsorbed in the distal convoluted tubule, and if there's parathyroid hormone present, then it will open channels in the apical or luminal membrane that will allow calcium to come into these cells. The last portion of our tubular system is going to be the collecting ducts, and so collecting ducts will actually be responsible for reabsorbing some sodium in the presence of aldosterone, some chloride, some co-transport of hydrogen chloride and bicarbonate. Potassium is both reabsorbed and secreted here. That's going to be dependent on how much aldosterone is being produced, but this usually results in net potassium secretion. Water here can be brought in by osmosis, and this gets controlled by antidiuretic hormone, which inserts channels into the collecting duct to bring water back in. We'll examine that in more detail in the next video. And urea will come back in via facilitated diffusion. There's a concentration gradient in the deep medulla region that will bring urea, that will cause the reabsorption of urea. And again, we'll get into that in more detail in the next video. So what are the characteristics of normal urine that you have here? The volume can be 1 to 2 liters in 24 hours, and that kind of brings up a little bit of a funny story for me. At one of the last places where I worked down in Savannah, there was an extra credit assignment done by the lead A&P person. She would have her students in 24 hours pee into jugs to see how much urine that they were producing, and that was an extra credit exercise. So they had to take pictures of the containers. They didn't actually bring it into her. So the color of urine normally is going to be yellow or amber, but this varies with the concentration of the urine, and it also varies with the diet. The yellow color is usually due to urochrome, which is a pigment produced from the breakdown of bile, and urobilin from breakdown of hemoglobin. If the more concentrated that the urine is, the darker it is. There can be some things that you can get in the diet. Reddish colored urine can come from beets. Medications can affect it. Let's see, vitamin B can turn it this really bright yellow. And again, some pigments can pass straight on through. There is an old med school prank where people put methylene blue in coffee, and that will actually turn somebody's urine blue because methylene blue passes on through, it gets passed out through the urine. Not something that I would ever recommend any of you do. So urine is transparent when freshly voided, but can become cloudy upon standing. If when urine comes out at the time it's kind of cloudy, that can happen at night because some mucus will come out with it after you've slept at night, and this can turn the urine a little bit cloudy. Your pH is going to be between 4.6 and 8. It varies with diet. High protein diets increase acidity. Vegetarian diets increase alkalinity. And the specific gravity basically means how dense is it. So it ranges from just a little bit more dense than distilled water to a bit more than distilled water. And so if it weighs more, it basically means that it has more solutes in it.